Hey, it's Moser, and I want to talk with you about DNA technologies and compare the three technologies that you should be aware of. Well, first, let's talk about DNA, deoxyribonucleic acid. If you've ever watched CSI, you know that it can be used to solve crimes. If you've ever watched Jerry Springer, you know that it can be used to prove paternity. And it can be used for disease testing and to find your family. Well, how? How do we use DNA, this long, skinny molecule, to do all these amazing things? So there are three technologies that biologists, doctors, and crime fighters use to take DNA from just a molecule to information: DNA extraction, DNA gel electrophoresis, and DNA sequencing. DNA extraction is the simplest of these three, and it's also the first step in either of the other two. It doesn't provide a whole lot of information, though. DNA extraction really just proves that whatever it is you took this sample from has DNA, which just proves that it either is currently a living thing or was at one time a living thing. And this is super simple. You can do this at home. You can do it in your kitchen. You can take DNA and extract it from bananas, chicken livers, split peas, onions, your own mouth. It's really simple. All you need is some very simple tools, some rubbing alcohol and dish soap, a blender. But it doesn't give you a whole lot of information. It doesn't give you any information about the living thing the DNA came from. It doesn't tell you what species it is. It doesn't tell you what color hair it had or what color wrapper it had. To do that, you're going to need to go a step further. This is DNA gel electrophoresis. And once DNA has been extracted, that sample can be cleaned up, put into a special gel mold. It kind of looks like clear Jell-O. And then electrical current can be run through it. This separates the DNA into little strands of different lengths, and it allows you to make comparisons between individuals. So you can see how much DNA two individuals share. This is what is commonly used for paternity testing and for crime scene DNA comparisons. Now, gel electrophoresis doesn't give you any information about the characteristics of an individual. It just shows you how much DNA two samples have in common. So it is useful because we know how much DNA we expect parents and their offspring to share, 50%. We know how much DNA we expect siblings to share. So it's great for finding family. It's great for looking at, did this DNA that was left behind at a crime scene match this person? But it doesn't tell us if someone was 6'2" and had brown hair or, has a gene for a particular mutation. To do that, we have to go one step further. That would be full DNA sequencing. And in DNA sequencing, again, the DNA has to be extracted first and the sample cleaned up. Then the sample is put into a machine that actually shows every single base in the DNA sample. It shows individual genes that may be present. It shows mutations that may be present. And with a full DNA sequence, you can actually say, this is a human being with red hair, brown eyes, and a breast cancer gene. Whoa, this is a very high level of detail. Now, because this is the highest level of detail, it's also the most expensive and the most complicated and the least frequently used. So, quick review. Which of the three technologies that were discussed here provides the least information? That would be DNA extraction, because it just tells you that this is a living thing. Which of the three technologies that's discussed here is actually the first step to getting to the other two? Well, that would also be DNA extraction, because before you can run a sample through gel electrophoresis or do a full sequence on it, you have to extract it from whatever it was. Which of the three technologies discussed here provides a way to just compare DNA from two individuals to find out how much DNA they have in common? That would be DNA gel electrophoresis. And which of the three technologies discussed here can actually tell you what color hair, eyes, or skin an individual has? That would be a full DNA sequence. The most complicated and least often used, but the one that provides the most information. Okay, excellent. Go on and do what's next.